everyone. Thank you for coming this afternoon. It's my pleasure to introduce Dr. Paul Koch. Dr. Paul Koch is at the University of Wisconsin. He is you're born in Michigan, but you moved to Wisconsin when you were 10, and he's never left.
I've gone on vacation. I've traveled out of the state.
Well, yeah, that's true. But he's got all his degrees at Wisconsin, and they must like him there. They've kept him there for quite a long time. Paul is actually part of the evil empire of turfgrass pathology. I'm a part of that as well. So I went to grad school with Jim Kearns under Dr. Elaine Treadway, who was my advisor. And Paul went to school or was advised by Jim Kearns. So we're kind of in the same little turfgrass pathology tree, and they do call us the evil empire. I'm not sure what character I am though. Maybe a Wookiee, I'm not sure.
I don't really know Star Wars.
Yeah. All right. So with that, I'll let Paul take it away.
All right. Thank you, Lee, and thank you for the invitation to come, and thank you everyone for coming today. I know it's a busy time of year, and I appreciate the flexibility. I kind of drove in earlier today, and I'm gonna leave tomorrow. So I've got a 2 and a 4-year-old at home, so overnight travel is difficult. So I appreciate the flexibility and still being able to come down and meet with the department and talk about some of the work that we're doing at Wisconsin. So speaking of difficult, dollar spot, right? That's I'll tell my family I use them in my presentation today, talk about how difficult disease management is. So dollar spot, what is it with dollar spot? I don't know how many of you talked with Lee a lot, but he's probably mentioned dollar spot a lot. It's a significant disease for those of us in turfgrass pathology, probably the most significant disease if you go by the number of pesticide applications made in a single season. So this is what dollar spot looks like in a fairway situation. There's aesthetic issues with it. It impacts the playability of the golf course. And it requires a lot of fungicide applications. Depending on the situation, it can be 10 or more fungicide applications in a single season targeting just this particular disease. The reason for that is dollar spot, the pathogen that causes dollar spot, Claridia species. It's a complex of species. It's active at a very mild temperature range, from 65 to 85 degrees Fahrenheit. We have those temperatures for the vast majority of the summer and fall and spring throughout much of the Midwest. So it can really occur, at least in Wisconsin, we oftentimes see it starting in mid-May through early November in some years. So a very long period of time. And there's just not a lot of cultural practices that significantly reduce the activity of the disease. So really, we're very reliant on chemical fungicides to suppress this disease. However, we're starting to approach some difficulties with the repeated use of fungicides for dollar spot control. These products, the costs have increased significantly since COVID. We have widespread fungicide resistance to multiple chemical classes, the benzimidazoles, the DMIs, and most recently, the SDHI class of chemistries is kind of the newest class of chemistries. And I don't know about you, Lee, but up in Wisconsin, we are seeing a lot of very rapid development of resistance to individual SDHI chemistries. And we don't have new chemistries coming down the pipe at any great rate. And so we're really starting to see serious issues with resistance and fungicide resistance and dollar spot. And then, of course, there's regulatory, human health, environmental, social concerns. On the regulatory front, there has been a lot of activity with the EPA and restriction of the use of some of our fungicides that we use very commonly, very regularly. Chlorothalonil will likely have some severe restrictions that should be announced probably relatively soon. Iprodylon is another chemical that we oftentimes use for dollar spot control. Likely to be some significant restrictions there. So the future of chemical control for dollar spot is murky and certainly uncertain. And so a lot of the research that we're doing in my lab at Wisconsin is focused on understanding more about the pathogen, and then taking some of that knowledge about the pathogen and developing some more integrated alternative type control strategies. So a lot of this is stuff we're working on. So I know you like to come to these seminars and you get this very kind of complete idea and conclusions at the end of the talk. That's not really what I'm going to be able to show you today. I'm going to show you sort of our thought process based on what we know, what we've learned about the pathogen in the past few years, and sort of where we're going with some of that research. So keep that in mind as we go throughout the presentation today. All right, the first study that I want to talk about is kind of a wonky, very applied extension project. And that's the influence of nitrogen on dollar spot severity. So forever and ever, in decades and decades, dollar spot has been known as a low nitrogen disease, meaning the disease is more severe in low nitrogen situations. However, when you go back and look at what that actually means, and you go back and look at some of the research that's been done with nitrogen fertilization and dollar spot, most of that research was done in the 60s and the 70s and the 80s when they fertilized golf courses differently than they do today. They used to fertilize golf courses with 1 or 2 or 3 larger granular applications of fertilizer. How we fertilize golf courses now is through repeated applications of very light, oftentimes sprayable or liquid applied foliar fertilizers. And so there had been very little research looking at the impact of nitrogen fertilization applied in this spoon feeding approach. So I had a master's student, Ron Townsend, and he joined me back in 2016. We did this project just looking at the impact of various rates of nitrogen fertilizer, but applied in a spoon feeding approach through a liquid applied fertilizer. What was the impact on dollar spot? So here's a kind of a picture teaser. Dollar spot does have nitrogen does have an impact, right? So here's no nitrogen over the course of the year, and then here's 20 kilograms of nitrogen per hectare, which is about 6 pounds of N per 1,000 square feet, which is a lot of N, right? You would not apply that much N on a golf course. So I'm going to kind of skip over a lot of the basics of how we did the study. We looked at different rates, 0, 5, 10, 18, and then this 28 rate. And what we were expecting to see is a stepwise decrease in the amount of dollar spot with each increasing rate of nitrogen fertilizer. What we saw, the reality was much different. We saw basically no impact of nitrogen fertilizer at these low rates. We start to see a little bit of a wobble at this 4 pounds of nitrogen per 1,000 square feet, and then a huge drop once we get up to this really, really high rate of nitrogen. So when I give this presentation to golf course superintendents, you know, they leave the room saying, "Oh, Paul's recommending we apply 6 pounds of N per year." I am not. That's not the point of this study. This is too much nitrogen. The grass would be growing uncontrollably. If we were concerned about the environmental effects of repeated fungicides for dollar spot control, we would be more concerned about the nitrogen leaching at this high rate of nitrogen. So really, the point is what is the mechanism of this nitrogen, and why do we see this huge cutoff from this 4 pounds of N per 1,000 square feet up to the 6 pounds of N per 1,000 square feet? We also looked at foliar nitrogen content of the leaf blades. We did a study at 2 locations. One was at our research facility in Madison, and then the other was at North Shore Country Club on the north side of Chicago with Dan Dinelli, who is a superintendent, who's a very famous superintendent. He's very into research. He probably has more research projects on his golf course than most university research farms have at their station. And what we found was a very sharp cutoff in symptoms once nitrogen foliar content got above 4.5%. So we can take that information and move forward as we try to understand what the nitrogen is actually doing. Is there another way we can get to this 4.5% cutoff without applying that much N? That's kind of our thought process as we think throughout the impacts of nitrogen and how we can potentially get the benefits of all that nitrogen without applying that much nitrogen. That's kind of one of the goals we have as we were thinking about this project. Now, I'm going to show this slide here, and then I'm going to come back to it in a little bit. So we did that nitrogen study with Ron in 2016, 2015, 16, and 17. The summers of 15, 16, and 17. And then we ended the study. Ron got his master's degree, went off to work for Syngenta, where he's at now. And then we just let that plot go the next year. We didn't do anything to it except mow it and water it. And what we found in 2018 is that the pattern held. The high rate of nitrogen still had the lowest level of dollar spot the following year. But what was actually more interesting than that is that within this treatment set of treatments, we had a fungicide program to act as a positive control that also did not receive any fertilizer. And when we look at that plot the following year, the amount of disease that developed on that plot was much, much higher than any of the fungicide treated or the fertilizer treated plots, and also higher than the plot that had not received any nitrogen for 3 years. So something with that fungicide program is leading to more dollar spot the following year. Keep that one in the back of your mind. We'll come back to it in a little bit. All right, so if we go back to 2017, I am in my 3rd year as an assistant professor at the University of Wisconsin, and we are starting to get into some microbiome. And I brought in a postdoc, Michael Milliken. He came from Win Beatty's lab at Iowa State. And he was sort of getting a lot of our microbiome work up and running. And we thought that there might be a microbial component to that sharp cutoff between 4 and 6 pounds of N and the impact that it had on dollar spot development. So what Michael did is he went in 2017 and he sampled from the no nitrogen plot. He sampled from the 1 pound nitrogen treatment, which is labeled as low here. And then he also sampled from the high nitrogen treatment, which is the 6 pounds of N. And this is just showing the beta diversity here. And really all these dots are showing is these are each individual samples of just the bacteria in the soil. And the closer the dots are together, the more similar their bacterial communities are. And so prior to initiating the treatments in the 3rd year of the study, we didn't really see any pattern. They're kind of randomly scattered around. But when we look at after 2 months of those nitrogen fertilizer applications, the bacterial communities in the high nitrogen plot are becoming more similar than any of the bacterial communities in these other nitrogen plots. So that further strengthens our thought that there might be a microbial mechanism behind the nitrogen impacts. So we also looked at increased the relative abundance in these phylum, and we found a lot of changes in the proteobacteria in the high nitrogen samples. Now, for those of you who are bacteriologists, you're probably thinking, Paul, like every bacteria is a proteobacteria, right? There's going to be a whole bunch of different changes there, and that is true. But we did see a lot of changes within that proteobacteria phylum, and we know that there's a lot of well-documented biocontrol bacteria that exist within the proteobacteria phylum. So again, like I said before, I'm kind of walking you through our thought process over the period of several years. So we see that beta diversity, everything kind of the high nitrogen samples are more similar. Now we see more changes in proteobacteria relative to most other bacterial phylum in just the high nitrogen samples. So then we just started to do some other work. And oxalic acid, I'm going to talk a lot more about oxalic acid in a few slides, but oxalic acid has been proposed as an important virulence factor in dollar spot, much like it is in Sclerotinia sclerotiarum in soybeans. And so we found some of these papers looking at bacteria that can degrade oxalic acid produced by fungal pathogens. And there's this FRC gene, which is formal-CoA transferase, which can be has been demonstrated to be a biomarker for bacteria that degrade oxalic acid. So we also we had those samples that we had taken from the nitrogen plot, and we measured the expression of FRC in those individual plots, and there's markedly higher FRC expression in the high nitrogen plots, which to us now, as we're thinking through this thought process, if we are putting down a high amount of nitrogen, it's possible that there is a certain segment of that bacterial community that's very good at utilizing the nitrogen as a carbon source and increases in population rapidly. And part of that bacterial population is effective at degrading oxalic acid, which might prevent the fungal infection from occurring that causes dollar spot. That's kind of our hypothesis of why just that high rate of nitrogen works so well controlling dollar spot, but the other rates did not. So we have work that we're following up on. I have a PhD student, Dawen Huo, who is in her final year. She's actually going to be defending in July, but she's done a lot of follow-up work on this, and I'll talk about some of her work here in a minute, but there's other work that has identified specific bacteria that degrade oxalic acid. So part of Darwin's work was going back. We kind of recreated that nitrogen study on a new plot. We did that for 3 years, and Darwin went out to the plots and isolated individual bacterial strains that degraded oxalic acid. So we have about 15 strains that can degrade oxalic acid that we have in the freezer, and we put them in a plate and do a competition assay in the plate, and they can effectively suppress the growth of the dollar spot fungus. So potentially some targets for improved biocontrol for dollar spot control in turfgrass. Biocontrol of dollar spot and turfgrass has traditionally been poor, poor to be nice. All right. So I've mentioned oxalic acid. So let's reset here, kind of go in a bit of a different direction and focus on oxalic acid. So what is oxalic acid? It's a key virulence factor for some plant pathogens. For some plant pathogens, it's a pathogenicity factor. So if you knock out oxalic acid production in Sclerotinia sclerotiorum, it will not infect. Sclerotinia sclerotiorum causes white mold in soybeans. So if you knock that out in white mold, it will not infect. Other pathogens like botrytis, it appears to be more of a virulence factor. You still get disease, but you don't get as much disease as if you had the full production of oxalic acid. So there has been a fair amount of work looking at oxalic acid in turfgrass and dollar spot in particular. So Mike Bain's program at Ohio State, before he moved into administration, was looking at oxalic acid and dollar spot. Kind of showed an association. They'd see sort of increased levels of oxalic acid in the presence of dollar spot. One of my PhD mates in Jim Kern's lab, Renee Rios, she did some work looking at development of OA and production of OA in response to infection in various monocot hosts. So again, associations, but nothing real conclusive that oxalic acid was really important in dollar spot. And so again, one of the projects that Ron Townsend did, that Ron is the master's student who did that nitrogen work, he also did a fair amount of laboratory work with the dollar spot fungus, growing them at different pHs and measuring the amount of oxalic acid that the fungus produced. And so over here on the left is just what Ron did here is he grew the fungus at 2 different pHs in media, in a liquid media, PDB, at a pH of 7 and a pH of 4. And if he grew the pathogen at a pH of 4, the pH stayed consistent in the media throughout the entire process. But if he grew it at a pH of 7, over time, the pH decreased. And I don't have the data here, but the production of oxalic acid increased. We could measure more oxalic acid in that medium. In addition to that, he grew the dollar spot fungus on different solid media. So PDA amended 2 different pHs, pH of 4, pH of 7, and pH of 9. So at the pH of 4, the low pH, the dollar spot fungus grew most aggressively, grew fastest. At the higher pHs, it grew slower. So again, our thought process here is that if it's a high pH, the fungus does not like that high pH for infecting. It prefers a more acidic pH to infect the leaf. And this has been demonstrated in Sclerotinia sclerotiorum with white mold in soybeans. And so what we think is happening is there's a metabolic cost to the fungus to produce that OA to optimize the infection. So then, as an extension person, right, I'm cursed with never being able to just appreciate biology for what it is. We need to always take something and how can we manipulate it for control of the disease in the field, right? So we're thinking about how can we take this information and use it to control disease in the field. And so our thought then is, well, what if we can increase the pH of the leaf so much that we make the fungus work so hard to produce so much OA that there's not enough energy left over inside the fungus to actually infect and cause disease. So that is the project of Zoltan Banyai, and he's a master's student in my lab. He's going to be entering year number 2 this upcoming summer. And so what Zoltan was doing, what Zoltan's project is looking at different products that increase the pH of the leaf, different high pH products. So there's 2 different studies that Zoltan is doing. He's looking at different products that increase pH. So we went and talked to somebody in soil science, Phil Barak at the University of Wisconsin, about what we were trying to do. He's kind of a pH plant soil expert. And he gave us 3 or 4 different compounds that what we asked him for is like, Dr. Barak, we would like something that increases pH and doesn't kill the plant, right? That was our two questions for Phil. And he gave us 4 compounds, and we tested those. And then we had been doing some preliminary work with an individual compound, potassium bicarbonate, and had seen some success with that. And so then we were trying to optimize the use of that individual compound. It's kind of two similar, related, but slightly different studies. One thing that I won't talk about this later, so I'll show it now. There is some phyto with some of the products, right? That is what we're looking at here with some of this browning. It's a quick flash. It's gone within 2 or 3 days. Doesn't kill the turf, but it is a pretty noticeable phyto with some of these products for the first couple of days. All right, so here's the treatment list for looking at different products. So potassium bicarbonate, potassium carbonate, ethanolamine. Caligreen is a commercially available form of potassium bicarbonate. We were testing potassium bicarbonate at much higher rates than the Caligreen label. Then we have sulfuric acid in there. The point of the sulfuric acid treatment is if we can increase the pH to decrease dollar spot, if we lower the pH, would we make dollar spot worse? Right, by like pre-optimizing the infection on the leaf surface or the conditions on the leaf surface. And then the potassium sulfate is a pH neutral product. The reason that product is in there is we wanted to see if potassium was doing anything, right? We kind of wanted to rule out the impact of potassium itself, not the pH impact, which is really what our project is more interested in. So that's the project where we're looking at different types of products. Then we focus in on potassium bicarbonate in this other study because we had success with that product in the past, a couple of years ago. And here we're just looking at different reapplication intervals and different water volume applications. So the reapplication intervals were 7, 14, and 21 days. And then the water volumes were, these equate to, I believe, 0.75, 0.5, 1, and 2 gallons of water per 1,000 square feet, which are, 0.5 is a little on the low end. Most golf course superintendents are right about 1 gallon of water per 1,000 square feet when they're applying fungicides onto their turf. All right, so quick look at some of the results. This is just from last year, and these are slides that were produced by Zoltan for his talk at the American Society, the Crop Science Society of America presentation back in St. Louis in November. And what we find is that most of these products are very effective at controlling disease. This is area under the disease progress curve for dollar spot over the course of the entire season. And the higher rates in all of these cases were better than the lower rates. The Caligrain low rate was much less effective because it just has less potassium bicarbonate than what we're testing over here. And then the other question that we had, right, was here's the sulfuric acid. We don't see that it's causing any more disease than the non-treated. So kind of one of our hypotheses was whether if you acidify things, do you make dollar spot worse? This data doesn't support that. And then here's that potassium sulfate. No suppression of dollar spot with the potassium sulfate compared to any of the pH effects. So this matches up and supports previous research that has shown potassium on its own does not have an impact on dollar spot. But so a lot of different products that we can use that are providing significant control of dollar spot. Here's the one where we focus in on potassium bicarbonate and we look at the different reapplication intervals and the different water volumes, right? So the colors are the water volumes. So green is the lowest water volume, orange is the middle, purple is the higher one. We didn't see any impact of the water volume at all, right? So when we're looking at different water volumes to spray, we didn't really see any impact of those water volumes. The higher the water volume, the better the coverage and the deeper into the canopy that product gets. So it doesn't look like that's really having much of an effect. But the reapplication interval did have an effect. Here is 7, 14, and 21 days. As you might expect, the shorter the reapplication interval, the more efficacy that we saw. So the 7 days provided much better control than 14-day reapplication intervals, which provided much better control than the 21-day reapplication. And here's a picture showing some of the results, right? So this was we had a very weird year in Wisconsin, incredibly dry year in Wisconsin last year. So disease was very late in developing. We had the driest May and June on record for Madison, Wisconsin last year. Disease really didn't develop until end of July. So this was kind of a late developing epidemic. So typically by the end of August, we would have much more disease than this. But still plenty of disease there. And really the treatment plot here, this is potassium bicarbonate at a 7-day reapplication interval at 1 gallon per 1,000 square feet. There's a couple of small speckles if you look really hard in there, but overall we're seeing really, really good control using this product. So this study was also replicated down at Dan Dinelli's place on the north side of Chicago, and they had more dollar spot than we did. You can see this is how severe dollar spot can become in a golf course putting green situation. This would be an unplayable surface, right? The superintendent would lose their job if they had putting surfaces like this. Luckily, this is kind of a side nursery, and like I said before, Dan has more research than a lot of universities do going on at his golf course. So here's the potassium bicarbonate treatments. You can spot them very readily compared to some of the non-treatments. So moving forward, what do we want to do with this? I think we're not necessarily going to recommend that everybody go out and spray potassium bicarbonate. I don't think that's what we're doing. But we want to kind of show that pH does play a role in dollar spot. So what we can do is tell private industry, what are some ways that you can effectively increase leaf pH? Are there other products that you have? Because right now I'm not exactly sure how financially feasible regular applications of potassium bicarbonate would be. We purchase them scientifically. We purchase the scientific grade product. So if we purchased more commercial grade product, I don't know what the finances would look like, but through the scientific grade product, it's not financially feasible for a golf course. So I think this is more of an understanding the biology, showing that this is an effective way to suppress dollar spot. There are some modest phytotoxicity issues that we need to address. But this concept, I think we got another year to go with this upcoming year, but I think based on just the first year, can very significantly and effectively suppress dollar spot. And in particular, I think this is important information for other areas of the world that don't have as many fungicides as we do in the US, in particular Europe. So Europe, dollar spot was never an issue in Europe. And in the past 5 to 6 years, it's become a huge issue because of climate change and the higher humidities in Northern Europe and in particular Scandinavia. Dollar spot is becoming very severe. In Scandinavia, they don't have really any fungicides for use. So something like this, where even though it's not perfect, it's better to have this than this. And so that's where we're going with some of this. Okay, we have another year to go. We'll see what data we get. Okay, so I mentioned Dallin before. She's doing a lot of this OA work, and she really wanted to do a lot of molecular work. Her background, she had a master's degree from Ohio State in tomatoes, doing molecular work in tomatoes. And so she came to my lab, and we wanted to utilize her molecular expertise to really get a better understanding of what OA is doing within Claridia, the dollar spot pathogen. Okay, so what Dallin did is Dallin worked with Mehdi Koch, who's a professor in our department at Wisconsin, who does a lot of molecular plant pathology work. In his particular, his lab is adept at knocking out certain genes. And so she worked with Mehdi's lab and used CRISPR-Cas9 to identify and knock out particular oxalic acid producing genes in Claridia based on homologous genes in Sclerotinia flourisciorum. And so what she did, she created 2 different knockouts. We compare it to the wild type and then a different wild type. This LWC is an isolate of Claridia that's been fully sequenced. And so we used actually this isolate to determine which genes to knock out here. And so both at a low pH and at a higher pH, we would expect more oxalic acid to be produced. In both these situations, oxalic acid production is much, much lower. We are still producing a little bit of oxalic acid. We're still detecting, I guess, a little bit of oxalic acid. So it could be that there's likely maybe other genes that are involved in oxalic acid production. So there's still some oxalic acid production, but we're limiting significantly the development of oxalic acid in these knockout pathogens or in these mutated isolates. So then what does that How does that relate to disease development on the turf? Well, we saw a lot less dollar spot development in the turf. So these top two lines are the two isolates where we did not knock out the OA production genes, and these bottom two isolates were the ones where we did knock out the OA isolates. We're not exactly sure why we're seeing significant differences or nearly significant differences in the development of disease with those two strains, and that's something we're kind of exploring a little bit and further discussing. And we are still seeing disease. So that gets to the point of we're still seeing some oxalic acid production. We're still seeing some disease. So there's likely other things that are involved in dollar spot development in turf. So these ratings here are these are a digital image analysis of these pots. And so up on the top is we inoculated the pots with the different isolates. So these are the two rows that where the oxalic acid was not knocked out, and these are the two columns where the oxalic acid was knocked out. And we see the initial development of mycelium, but then over time, kind of go down here to the bottom, these two are much more brown, much more impacted by dollar spot compared to these two. So I think we are comfortable saying that oxalic acid definitely does have an important role in the development of dollar spot in turfgrass. Maybe not so much a pathogenicity factor like we see in sclerotinia, probably more similar to botrytis where it's a virulence factor, but certainly it does have an important role in the development of disease. So again, how do we use this information to control disease? There's kind of two approaches with double-stranded RNAs, or RNAi, RNA interference. And so one approach has been to breed transgenic plants that produce that double-stranded RNA that targets the gene in question and turns it off and suppresses development of disease. So in our case, it would be most likely a bentgrass cultivar that has been transgenically developed that produces the double-stranded RNAs to target the oxalic acid gene and turn it off. However, transgenic grasses are just not really a thing. And we only replant golf courses once every 20 years, if that. There's golf courses out there that are 100 years and older. So this is probably not going to be the most effective way to incorporate RNAi technology into turfgrass. So what we have targeted is applying the double-stranded RNA molecules as an exogenous product, an exogenously sprayed product, really is like a biofungicide that would then be absorbed by the fungus and turn off that OA production and hopefully limit the development of disease. And so again, our collaborator, Mehdi Kabaj, has done this work in soybeans. And so this is from a paper that Mehdi's lab produced. Now, this was a plant where they transgenically created the plant to produce the double-stranded RNAs endogenously. So they are produced by the plant, and they lead to a significant reduction in the amount of white mold that occurs. So really for us, what we want to do though is again, focus on those exogenous applications. So step one was making sure that the Claridia, the dollar spot fungus, can take up and absorb the product, which it has. We've confirmed that using electron microscopy that it will take up the double-stranded RNA product. Now the next step is applying the product as a spray on the turf, inoculating with the dollar spot fungus, and seeing how much disease develops. And so we're actually doing this in the growth chamber right now. Last week is when Darwin did the first round of this exogenous application of double-stranded RNAs. And so I just looked at it with her 2 days ago before I came down here, and we still need to figure out exactly the right concentration of double-stranded RNAs. The other issue that's very common with double-stranded RNAs is they break down very quickly. So we're going to try and look at some ways to increase the efficacy. That's all to say that it didn't look like it really worked very well the other day when I looked at the plants with Darwin. So, but that was our first try, kind of taking a stab in the dark with concentrations. We're going to increase the concentration of the double-stranded RNAs and repeat this, and hopefully we get to a point where we're having better success. We're also we're working with the Kabaj lab to produce additional double-stranded RNAs targeting other diseases and mix those products together, kind of like a multi-site fungicide would work. Okay, Got a few minutes left. I'm going to switch gears a little bit. I mentioned some of our microbiome work before. We have a bunch of different projects. You know, we kind of spent the first few years kind of wandering through this microbiome space, spending a lot of money, and really have kind of focused on the impacts of pesticides on the microbiome. So I'm going to talk about one additional study that we've done, and it comes back to this slide that I showed you before, right? So here we had these different rates of nitrogen that were impacting dollar spot, but what we noticed is the year after we sprayed this fungicide program, the dollar spot was much more severe in that plot compared to any of the other plots that had never received fungicide over the previous 3 years. So our thought process, again, it's a big thought process talk today, is that when the fungicides are present, they are suppressing the pathogen, but they might also be damaging surrounding competing microbes or antagonistic microbes, so that when you remove the fungicide that is suppressing the pathogen, there's not as much competition, and the pathogen could come back faster and stronger than before, right? A concept that's very well established in entomology, but is not as well established in plant pathology. So we have this, we did a 2-year preliminary study, and then last year we got a USDA agricultural microbiomes grant to study this in more detail. So this concept of what we call the disease resurgence or disease rebound, we're studying in much more detail and collaborating with metabolomics experts, and we hope to have much more information on what's going on here and what's going on with the microbiome and the metabolomics and all that process in a couple of years. But a different study that I want to talk about is kind of related, is this dollar spot suppressive soils project. Now, suppressive soils have not really been identified in any great degree in turfgrass, and certainly not golf courses. But we wanted to take our knowledge that we gained from that previous slide that I showed, and rather than apply pesticides and see how the microbiome reacts, we wanted to go out to golf courses that have been applying pesticides for years and years and see how their microbiomes differed. And so here's kind of the map of how we did it. So this was Ming-Yi Chow's project when he was a postdoc with me at Wisconsin. We chose 4 golf courses from the Midwest, from Wisconsin and Illinois. 2 of those golf courses had what I would consider a traditional fungicide program, where they sprayed on a very rigid calendar basis. If it was, you know, July 15th, they sprayed this chemical no matter what. Whereas the other 2 golf courses were kind of lower input. They didn't spray very often. When they did spray, they used kind of alternative products like iron sulfate or something like that. And we had 2 other ones from the Northeast. So 2 high kind of sort of high fungicide use and 2 low fungicide use golf courses from the Northeast. And then we also had some non-turfgrass places that we sampled: prairie, a corn site, and then a forest site in the Midwest. And then in the Northeast, a prairie site, an apple orchard, and then a forest site from the Northeast part of the country. So we had these samples mailed to us. This was supported by the Golf Course Superintendents Association, and we didn't have travel money in the budget, so they took the samples and they mailed them to us. And then what we did is we took out a small subset of the soil, and we transferred it to a sterile sand medium, and then kind of mixed it up. The thought process being is we want to take out the complicating factor of soil type and organic matter and all that, and just take the microbes and put them into a sterile medium. And then on top of that, we planted grass. We planted creeping bed grass. And then after a period of time, as that grass filled in, we inoculated with the dollar spot fungus, and we assessed how much disease developed. So really, the only thing that's different about these pots should be the microbes in the soil. Okay. So this is a busy graph, right? If I was a grad student, if I was a professor talking to my grad student about this, I would say this graph is too busy, but I'm still showing it to you because it's all here on one graph. So what this graph is these are all the different sites that we sampled. Okay, and so this is day 0. And what's a little bit backwards is that we did a digital image analysis on the percent greenness. So the lower the number, the less green, meaning the more disease. So the lower numbers have more disease. The higher numbers have less disease. So when we look at where the different golf course samples and non-golf course samples lined up here, it was really interesting. This is this green one that's kind of under this tab right here is one of the low fungicide use golf courses from the Midwest. And the other one, this blue one right here, is from the low fungicide usage golf course from the Northeast. Both had the highest levels of susceptibility, or the highest levels of resistance, excuse me. We also, here are a lot of the high-end, high fungicide usage, much less suppressive, more susceptible to the development of disease. What was also interesting, this one right here, what's the worst one? That's actually the control, the one that we didn't transfer any microbes over to that one. Right, so there's no microbes transferred. That one has the most amount of disease, likely due to no competition or minimal competition from any microbes. If we look at what is this one, right? This one is a prairie one from the Northeast. So had probably never been exposed to any pesticides, and this one is actually one of the prairie Midwestern sites, but it probably hadn't had much dollar spot development in a prairie situation before. So there probably wasn't much antagonism directly developed for the dollar spot pathogen in those systems. So really what our hypothesis up here is that very few pesticides applied on these sites, plenty of exposure to the dollar spot fungus, and so that over time it builds up a suppressive microbial population. And this is kind of the difference. This is what one of the more resistant pots look like, and here is one of the more susceptible pots, the development of dollar spot in those pots. Okay, so Ming Yu also went in and we got the fungicide records from the golf course superintendents that also submitted samples. And there is nothing that angers a golf course superintendent as much as asking for their chemical records, but they all eventually gave their records to us. And what we just did is we correlated the frequency of fungicide use and the number of applications per year with the amount of disease that we saw in our assays. And it was a very clear result that the fewer fungicides that were used, the less disease that we saw in those situations. Ming has since done a follow-up analysis, and he actually correlated the weight of fungicide active ingredients with the amount of disease, and he found a very specific number of a specific weight of AI, where if you were above that number, you had more disease, and if you were below that number, you had less disease. And I'm forgetting what that number is now. He just told me about it last week, and I forgot to enter it in on the slide. But very specific numbers. Now, caveat here, this is based on 8 golf courses. There are thousands across the country. So, but I think this shows, these are golf courses from very different parts of the country, and we're seeing a very similar response. So what would we tell a golf course superintendent from this project? Well, we're not going to go out and tell them not to spray, because they wouldn't respond very well to that. But what I think what we could say is that you want to target your sprays, right? We're not saying don't spray at all, but what we are saying is that you can spray certain products, or you can spray too much that damage the competition. So you're really doing yourself a disservice and knocking back the health, the microbial health that's in that system if you spray too much. So we do some more work to kind of figure out exactly what is too much and what are the products that are most damaging to the competing organisms in the soil. And that's part of the USDA Ag Microbiomes project, is assessing individual chemistries and seeing which ones we see the biggest effects from those individual chemistries. So that's what we have doing. So hopefully we have some more information on that in a few years. All right, just a couple thoughts here. I talked before, chemical control options are declining. They will continue to decline most likely. We're going to need to get better at integrating all of our options as plant managers, whether you're in ag or fruits or hort or us in turf. We're just going to need to get better at using all this suite of tools for more effectively controlling disease, because we're going to be less reliant on them. And then I'm a field-based person. I recognize that, but I know the importance of some of these molecular tools. And so we really work with a lot of really smart, really good people in our department, and brought these people into my lab at times to really integrate some of these molecular approaches to help us understand the system and develop some of these more integrated approaches. So with that, that's all the information that I have. Here's my contact information. Of course, here's my lab, right? Kurt Hockmeyer, who is a Purdue grad. He's a Purdue undergrad and Purdue master's grad, got his master's with Rick Latin. And Kurt runs my field fungicide program and field research program, and also manages our turfgrass diagnostic lab at the University of Wisconsin. And he's an incredible asset to my program and all my grad students and undergrads that work in the lab. They're, they do the work that allows me to come do these fun things and present these things to you. So I'm very grateful for their help. So with that, thank you for attending, and I'm happy to answer any questions that anybody has.
Question about potassium bicarbonate. Even if it isn't a product that you might recommend for usage, even after your study, would you be looking at how that might affect other pathogens on turf? Because if you're looking at it in one situation, whether it was used, or at least looking at these pH changes in other systems, would you see a change there as well? Is that something you might be interested in?
Yeah, so I mean, so a lot of these things that we're looking at are dollar spot specific. And dollar spot is not the only disease that occurs in turfgrass. And so we definitely need to get a better feel for how these different practices also influence other diseases. Because if we just control dollar spot, we don't control other aspects, we're still gonna have those other diseases will become more important. So I don't know if that answered your question or not, but I mean, at some point we need to look at what impacts these are having on other diseases as well. Something we haven't done yet.
So a question about disease assessment and measurement. So all of these is critical for you to look at the data and see the trends, etc. So my limited experience on these. When I see visual assessments on dollar spot, so I usually see peaks, valleys, peaks and valleys.
Mm-hmm.
I'm assuming that that's because there's a mowing process to get rid of disease tissue.
Mm-hmm.
But when I see the percent greenness, that's a more smooth curve that you're showing.
Mm-hmm.
I guess the question is, so perhaps bias towards imaging will provide a better curve, better assessment. But what are your thoughts about it? So I guess the question is, what would be preferable in the future, visual assessments or imaging?
Yep. So, well, I mean, it would be preferable to have imaging. I think part of the reason the imaging work that was done was, the percent greenness was not only was it all imaging, but it was also all in the growth chamber. Whereas the peaks and valleys, that tends to occur more in the field just because environmental conditions change. And as you said, we're mowing things. It's a very fluid environment out in the field. Whereas in a growth chamber, it's a much more consistent environment. So my guess is if we had taken digital image analysis of plants in the field, we would see peaks and valleys as well. As the environment changed, it becomes less humid, the pathogen becomes less active, the turf recovers over a period of 7 to 10 days, then humidity returns, the infection increases, disease increases, and we see the greenness decrease over time. So certainly the digital image analysis would take out a lot of that rate or bias. One of the problems, and I don't know, have you done much work with image analysis in dollar spot in the fields? Okay. One of the issues that we've had with digital image analysis with dollar spot in particular is that it can sometimes be tricky to differentiate dollar spot from other brownness in the field. And so like with certain diseases, snow mold, which I also do a ton of work on, it's much, in our experience, it's much more effective at differentiating snow mold because the line between disease and non-disease is so distinct. And with dollar spot, it's in some cases, it's less distinct. So there are other issues like scalping or drought or phytotoxicity that can cause browning in a summer environment, and it's more difficult to differentiate between those and dollar spot in the field. So that's an issue that we've had, we haven't approached and really worked on it a lot. I know other people have really worked with digital image analysis and dollar spot much more than we have, but that's one of the issues that we've had with doing digital image analysis in the field.
Follow-up question. So the ABCC values that you show there, I think there was a figure that showed 15,000 as a value. Another figure was 6,000. So there's variation for sure, but
Mhm.
for that, a follow-up question what that I asked to Lee the other day. So for a, let's say a 21-day period under the best conditions. So is that what you're expecting such values that are pretty high, 15,000
Yep.
over time, 10,000, 6,000, etc. What specific conditions are associated to that?
So like environmental conditions.
Yeah.
Yep, so, so to back up for a second, you know, I showed AUDPC here because of the simplicity and which is why we always use, you know, why we always, use AUDPC. But certainly we saw peaks and valleys throughout the course of the season. So it's not like, you know, that AUDPC value made it look like there was really no disease that developed. That wasn't the case. We did have peaks of disease that did occur, throughout the year. And when we see disease in Wisconsin and when most places see significant dollar spot development, it's due to high humidity. And so high humidity and mild warm temperatures. So overnight lows, upper 60s, daytime highs, mid to upper 80s. That's perfect weather. So when we have stretches like that for 3, 4, 5 days, we know we'll have a pretty significant outbreak of dollar spot. Yeah.
I have a question. If you could clarify low nitrogen diseases and how we understand them. Is it something that is like the plant is stressed due to nitrogen deficiency and the pathogen isn't like opportunistic? Or is it that the low nitrogen enables the pathogen to infect, kind of like how low pH does?
Yeah. So nitrogen in turf is different than nitrogen in agronomic crops because we're not measuring yield, right? So I talked with Doug Soldat is a fertility specialist in turf at Wisconsin. So I'm good friends with Doug. We talk all the time. And I've seen a lot of his presentations. And he likes to say that turf is always nitrogen stressed when we grow it as a turf grass. If you were a farmer growing turf for yield, you would apply something like, I think Doug has calculated it, you would apply like 18 or 20 or 24 pounds of N per year if you wanted to maximize the amount of yield. So really, we're always in some level of nitrogen stress in a turf grass situation. However, I think it's, you know, it's typically been thought of as more of a host response, that the host is deficient in some defense activity, not really having an effect directly on the pathogen. Now, what we show here today is that maybe actually some of that impact is on the microbes, not necessarily the host plant itself.
Okay, so you were talking about the biocontrol and, so I know like biocontrol isn't super feasible because if you shove a random microbe into turf, it's likely gonna die because there's so much competition.
Yep.
I think. So you found that there was there were microbes that suppressed the OA production, which suppressed disease. So were you using that more of a way to confirm that OA is a virulence factor? And then would look into other ways of suppressing that and not necessarily microbes?
Well, no, both, really, what we were trying to do is we wanted to see if that was, we wanted to look at the mechanistic aspects of that. But we also wanted to see if we developed a microbe that was specific for dollar spot. There was a specific biocontrol for dollar spot, would that be more effective? Right? We're not necessarily looking at a microbe that would, you know, crowd out and colonize the surface of the leaf blade that would then fend off dollar spot. We know that's not likely to happen. But if we created if we isolated and found a specific microbe that was very targeted in how it interacted with the fungus and suppressed that oxalic acid, would that be more effective? Or could we, you know, isolate the compound that the bacteria is producing and develop that as sort of a bio-derived fungicide? So those are some of the things that we're thinking with the isolation of those molecules.
So do you have obviously a lot of microbiome data? Would you look at like indicator species then to like see like what possibilities there were? Like comparing, you know, high disease pressure, low disease pressure, and seeing the differences, looking at indicator species and then try to like use those? Or would you just like use subculturing techniques and isolations?
Yeah, the latter. we have a lot of microbiome data. What we did though is what Dowling did is she used a specific media that is designed to detect OA degradation. It's a cloudy media, and when you find an isolate that degrades oxalic acid, it clears out a zone of the zone around the isolate. So these are all the ones that we identified were culturable bacteria, which of course are a very small percentage of the overall bacterial in the system, but that's how we did. We did not look at the microbiome data for individual species. We looked at which ones were clearing out the OA on the media, then we isolated those and grew them out, purified them, and then we grew them in competition assays with the dollar spot fungus on other plates. And then I think we came with like 15 isolates that could degrade oxalic acid and inhibit the growth of the fungus. So it was all culture based, old school.
I think we have a couple more minutes. Any more questions? I have 20, but I'm gonna save those for later at dinner.
So, I'm just curious about using transgenic as a biocontrol. Are you, with turfgrass, are you allowed to use that legislatively? Like, I'm just thinking, we're so fussy about using transgenic.
Yeah.
field-
That was approved, right? Roundup Ready bluegrass was finally approved, I believe.
Oh, out now?
I think so. I believe there is a Roundup Ready Kentucky bluegrass that has been approved. It took decades. But I think it was approved within the past 5 years. I don't know of anybody that's planted it.
I planted it and killed it twice because they told me I had to.
Oh, I believe it's for, I don't know if they're actually selling it, but I believe it has been approved. But of course, the issue then is, if you are there's just a lot of like practical issues for that being out
I
there.
never thought of it.
Yeah. Yeah, no, it exists. It exists, and I believe, because it was created, gosh, probably 15, 20 years ago, maybe more than that.
Yeah.
And then it was just the concern from the USDA, whoever governs that kind of thing, was the cross pollination and creating, super weeds and that sort of thing. And so it wasn't available for forever. And then I thought that it was approved. Maybe I misread that.
I've not heard that.
Okay.
Is it the Provista?
Yes.
Yeah,
Yeah,
I think it's out.
yeah, I
But-
think it's out as well.
Didn't they do a warm season like a St. Augustine or a centipede grass? Same way?
Yeah, I'm not sure about the
Think
worms
so.
inside. Yeah, and talking with like, who would actually use it, like lawn care applicators, it would just be, there would be a lot of difficulties with trying to like manage, having which clientele has the Roundup Ready versus which ones don't, and, that sort of thing. But yeah, I believe it is available. That one thing, nothing else, but that one. All right, thanks,
All right.
everybody. Oh.